In this lesson, we are going to look at glycoproteins and their functions. In the comment section, I have shared a video on glycoproteins and glycosylation. So, the first example of glycoproteins is erythropoietin. Erythropoietin is a glycoprotein that is secreted by the kidney tissues. It consists of 165 amino acid residues. So out of these 165 amino acid residues, 3 asparagines are N-glycosylated, while 1 serine is O-glycosylated. Erythropoietin is a highly glycosylated hormone, and this helps it to be retained in the blood plasma without being easily cleared by the kidney tissues. The role of erythropoietin is to stimulate the secretion of red blood cells in the body. Secondly, let's look at mucin. Mucin refers to that glycoprotein that is normally secreted by the epithelial cells. It is a highly glycosylated hormone. Mucin contains a lot of threonine and serine residues. So due to this, it means it contains a lot of O-glycosylation. Mucin has got the ability to retain a lot of water molecules, and it is highly resistant to proteolysis. What makes it to retain a lot of water molecules is its high glycosylation. Majority of mucin is excreted in the extracellular matrix, although part of it sticks on the plasma membrane. The role of mucin is to form mucus barriers that lubricate and protect the epithelial tissues. Next is a tissue factor. A tissue factor refers to this hormone that is normally exposed during trauma. Exposure of a tissue factor in turn activates the extrinsic pathway, which in turn activates the blood clotting cascade. Lastly is immunoglobulin antibodies. These ones are used by our body's immune system to fight pathogens. We go through our notes. Erythropoietin. So this is produced by the kidney. It consists of 165 amino acid residues. In each of the residues, 3 asparagine residues are N-glycosylated, while 1 serine is O-glycosylated. Erythropoietin is highly glycosylated, and this helps to retain it in blood plasma without being easily cleared by the kidney. It is a hormone that stimulates production of red blood cells in the body. Mucin. It's a highly glycosylated hormone produced by epithelial cells. It consists of a higher number of serine and threonine residues that can form a higher number of O-glycosylation linkages. It is highly resistant to proteolysis and has a high ability to absorb water molecules due to its high glycosylation. Majority are released into extracellular matrix, although some stick to the plasma membrane. They form mucus barriers that lubricate and protect epithelial cells. Tissue factor. A glycoprotein exposed during trauma, its exposure activates the extrinsic pathway, which in turn activates blood clotting cascade. Next is immunoglobulin antibodies, which are used by our immune system to fight pathogens.